Hello everyone. Today we are going to understand jaundice, what it is, how it happens in the body, and how to differentiate its type in a very simple way. First, let's quickly revise normal bilirubin metabolism. When red blood cells break down, hemoglobin is released. Hemoglobin is split into heme and globin. Globin is reused, while heme is converted into biliverdin, and biliverdin is then reduced to bilirubin. This bilirubin is called indirect or unconjugated bilirubin. It is not water soluble, so it binds to albumin in the blood and is transported to the liver. Inside the liver cells, this indirect bilirubin gets conjugated with glucuronic acid to form direct bilirubin, which is water soluble. This direct bilirubin is excreted into bile, enters the intestine, and is finally eliminated. And this is normal bilirubin metabolism. Now, let's talk about hepatocellular jaundice. In hepatocellular jaundice, the liver cells themselves are damaged. The common causes of this jaundice are viral hepatitis, especially hepatitis A and E, alcoholic liver disease, drug-induced liver injury, or cirrhosis. So what happens here? Because the liver cells are damaged, their cell membranes lose integrity, the bile canaliculi are disrupted, and the transport functions of the liver decrease. As a result, the uptake of indirect bilirubin decreases. So, indirect bilirubin increases in blood. Some bilirubin still gets conjugated, forming direct bilirubin. But this direct bilirubin cannot be properly excreted into bile. Why? Because biliary secretion requires intact cell membranes, cytoskeleton, and tight junctions. So, instead of going into bile, direct bilirubin regurgitates back into the blood. That's why in hepatocellular jaundice, both indirect and direct bilirubin are increased. Lab findings you should remember. ALT and AST are markedly increased. Albumin may be low. Total protein is usually normal. A to G ratio is reversed. Serum alkaline phosphatase is only mildly increased. Prothrombin time is prolonged and is not correct with vitamin K. Next, obstructive jaundice. As the name suggests, obstructive jaundice occurs due to blockage of bile flow. The common causes of this jaundice are gallstones blocking the bile duct tumors, pancreatic head tumors. So, what happens here? In obstructive jaundice, the liver cells are normal. They can take up and conjugate bilirubin normally, but the problem is bile cannot flow into the intestine. So, direct bilirubin increases significantly because it regurgitates back into the blood. Indirect bilirubin is usually normal or maybe mildly increased due to back pressure effects. Laboratory findings. Serum alkaline phosphatase rises markedly. This is the key finding. ALT and AST are only mildly increased. Albumin is normal. A to G ratio is normal. Prothrombin time corrects with parenteral vitamin K. Finally, hemolytic jaundice. Hemolytic jaundice occurs due to excessive destruction of red blood cells. The common cause of this jaundice are malaria, hemolytic anemia, transfusion reactions, internal hemorrhage. So, what happens? Because too many RBCs are breaking down, indirect bilirubin increases. The liver is normal, but it gets overloaded. Lab findings: indirect bilirubin increased, direct bilirubin normal or slightly increased. ALT and AST are normal, alkaline phosphatase is normal, albumin normal. Prothrombin time is also normal. So, in this video, we'll learn what jaundice is, how bilirubin is formed and metabolized in the body, and most importantly, how to differentiate between hemolytic, hepatocellular, and obstructive jaundice based on their pathophysiology and laboratory findings.